start. So hello everyone and welcome to our webinar. I'm Emily Crossley, co-founder of Duchenne UK and mum to Eli with Duchenne. I'm also joined by Alex Johnson, co-founder of Duchenne UK and mum to Jack and founder of Joining Jack. So today's webinar is focusing on gene therapy as a potential treatment for Duchenne muscular dystrophy. And for us to be addressing this issue really feels like a big moment. When Alex and I's sons were diagnosed, we were told to forget gene therapy, that it wouldn't happen in their lifetimes. Well, here we are today. Our sons are 12 and 13, and we have not one but 4 companies developing gene therapy for DMD. So there is understandably a lot of excitement, but this really is pioneering science, and there are inevitably huge challenges. And today we're going to be focusing our webinar on one of those, the issue of immunity. On Monday, Duchenne UK announced that it had granted £500,000 to one of the world's leading gene therapy teams focusing on gene therapy and immunology. And we're thrilled to be joined today by Doctors Manuela Corti and Byrne from the Powell Gene Therapy Center at the University of Florida. So I'm going to turn first to our Director of Research, Dr. David Bull, who's going to give a very brief summary of the current landscape in gene therapy, and then on to Drs. Corti and Byrne to explain the challenges of immunity and what their project will address. So over to you, David.
Thanks, Emily, and hello, everyone. Yeah, as Emily says, I've been asked to do a brief gene therapy scene setting session so that we can make sure that everyone has the same background understanding, so that when Manuela and Barry talk about the new project, we're all going to be have that same level of understanding with them. I've just got the 2 slides, and the first one up now is actually going to take you through the basics of gene therapy. And as you know, genes are found or located at various points along the whole length of the DNA sequence, the famous double helix ladder, which you can see spinning away there on the left. Each rung of that ladder is made up of 2 of just 4 different molecules. And it's the specific order of these molecules in the rungs which make up our genes. And reading this order provides the instructions for the cell to make all of the specific proteins that the body needs. Now, in Duchenne muscular dystrophy, 1 or more elements, which we call exons, of the gene for dystrophin have been corrupted or duplicated, or they're missing. And the end result is that the cell machinery can no longer make the protein dystrophin. Now, scientists have for a long while been able to create perfect copies of genes, including the dystrophin gene, in a test tube. But for it to be useful as a treatment, we need to find a way of getting that new gene into the muscle cells so that they can make dystrophin. And it really is not easy. We can't just take a tablet of artificial DNA in the same way that we would a tablet of paracetamol, because the stomach will destroy it before it gets anywhere near a muscle. And we can't inject it generally either, because the body will see it as foreign and remove it. And anyway, we couldn't really get enough to the muscles in the first place. But what we now realize is that we have a potential natural helper, because viruses have over millions of years perfected a way of getting into cells and depositing their own, usually harmful, DNA. So if we could find a relatively innocuous virus, and in this case, the adeno-associated virus, you'll see it called AAV, if we could remove its DNA and replace it with a new copy of a dystrophin gene, then this modified virus could be injected and do what viruses do, and insert the new DNA into the cells. Now, that in principle is straightforward, but the technical challenges are absolutely huge. So for example, the large size of the dystrophin gene, it just simply won't fit inside the AAV. And we only want the virus, as I say, to target muscle cells, so we've got to find a way of doing that. And then we come on to the huge problem of the virus itself, and the focus of today's webinar, because the body will fight it if you put the virus in there. There will be an immune response, and the target of the project that we're working on is aimed at mitigating that. So before I hand over to Manuela, what I wanted to do on this next slide, please, Molly, is just to give a brief update of the clinical trials that are currently in progress. I've just listed the 3 that are key there at the moment. And what I want to say right from the outset, it really is important to note that all of these trials are focused on safety and tolerability. They're phase 1/2A trials. These sort of trials, you'll hear them called first time in human. And that tells you what you need to know. The first time in human, the primary aim is it safe? However, it does seem to make sense to look for secondary desirable effects too, such as, does the treatment produce dystrophin? And can we detect a positive impact on boys' functional ability? That latter one, the functional piece, I have to say is tricky with the low numbers at the moment, but we can get a sense. Excuse me. So let's look at Sarepta first. If you look in that second column, you'll see it says vector, AAV RH74. So Sarepta, unlike the other two, Pfizer and Solid, it does use the AAV, but it uses a slightly different subtype. So worth bearing this in mind. In terms of their work, currently, there's just been a publication of the 4 boys that have reached the 12-month post-administration phase. Well, what we can say is that the infusion of the virus is well tolerated, and indeed longer term, they have reported no serious adverse events. Although some mild and moderate ones have been reported in these trials, we can say though that the boys were recovered well from this and were cared for, but there were no serious adverse events. Then, in terms of the secondaries, they did detect microdystrophin. And in fact, they've detected at quite high levels, I mean, sometimes up to 70, 80%. Though we should say at the moment, we actually don't know what percentage microdystrophin we need. But it's clear the virus is doing its job. Microdystrophin protein is being produced. And interestingly, they have got some North Star measures, which they say are improved. Which is excellent news, but as I say, the numbers are low at the moment, but it's looking good. And of course, the other big thing that we're interested in, that the CK levels are coming down. Sarepta say that by the time they get to the end of 2022, all 41 boys will have completed this trial and will have a complete safety profile on their method, and will have a good idea of some of the clinical desirable effects. If we move to Pfizer, you'll notice the vector there, it's still an AAV, but it's AAV9, perhaps more commonly used. Currently, Pfizer have 9 boys in the phase 1B that have reported well, and again, there are no problems, no apparent problems with the infusion. And it's worth just mentioning, in the very, very early days of exon skipping, one of the really big worries was quite serious abrasions at the site of injection, but this is not the case. It appears to be very well tolerated. However, we have to say that Pfizer have reported 3 serious adverse events, 2 of which reflect complement activation, i.e. in other words, that this is part of the immune response. However, those safety events were manageable. All the boys are doing well, cared for, and remain on trial. Pfizer also reports similar encouraging efficacy in that they have sustained minidystrophin production, and they also see improvements in North Star at 12 months. Also, we just want to say about Pfizer, it's clearly one of the world's largest pharmaceutical companies, and they have been pouring in resources for a long time into large-scale manufacture. It's not just for Duchenne muscular dystrophy. So they are putting themselves in a good position such that when and if these trials are really effective, they have some of the capacity to get the new medicine out there. To finish off, I just want to briefly mention Solid Bioscience. Solid and Duchenne UK have quite a history together. We've worked together, we've supported them, and they've done a lot of lovely project work for us. At the moment, their trial, and it is again an AAV9, their trial remains on a clinical hold requested by the FDA. They're working very hard. It would appear that, as I say, it's an AAV9, the same as Pfizer, but they're clearly getting some serious adverse events. They believe that this relates to perhaps to the manufacturing process, and that's actually the focus of their activity. Having said that, in the boys that have received it, they've got some functional analysis, which does show that microdystrophin is there. It's there at much lower levels, but again, we don't know how much. But importantly, I think with Solid's work, it's very clear that the microdystrophin is localized appropriately where you'd expect to see it in the muscle cell membranes, and also that it's linked to one of the important enzyme proteins involved in the muscle function, and it is linking up with that. I'm going to stick there, leave it with those three. I'm now going to hand over to Dr. Manuela Corti, and she's going to explain how they're going to work to try to seek to mitigate some of these immune issues to see if we can improve the effectiveness of the gene therapy. So Manuela, I'll hand over to you if that's okay.
Thank you. So this was a great introduction, David. Thank you so much for giving this overview, which introduced very well this topic. And also I would like to thank Duchenne UK for supporting this very important project. So as you can see from what Dave presented, there are lots of aspects that we don't understand. Why do we have all these adverse events, and why there is variability between these trials? So we think that the answer of this problem is relating to immunoresponses to the AAV, the capsid that we are using to introduce dystrophin inside the body. So the objective of this, the first objective of this project that Duchenne UK is supporting, focus on understanding how we can safely administer AAV without or preventing these immunoresponses. And also the second aspect is how we can introduce multiple doses of the same AAV in the same person. And this has two, this aspect is very important for twofold. First of all, because there is somatic growth in a child, so we will expect that the treatment will be diluted throughout the year. So we really want to make sure that we are able to do multiple doses, but also because our hypothesis is that we can reach that very high dose with multiple lower doses and reduce the adverse events to the exposure. So we are building this project through other projects we conducted for different conditions such as Pompe disease, Canavan disease. So we already use an immunomodulation to prevent the immunoresponses. And this specific project is applied to DMD. So we are comparing the immunomodulation that we already know that is working, that we have been using in other conditions, to other new agents that might be safer. And we are applying this approach in a mouse model of DMD, and the next step will be in primates, with the purpose to prevent these immunoresponses to AAV. We really think that these adverse events we found in the other study are relating to the formation of antibody, and the immunosuppression will prevent the formation of antibody and therefore allow for a safe administration and a safe treatment. If you go to the next slide, the second objective of this of our project is to focus on a very important challenge. As you might know, lots of people with DMD will be excluded by trials due to their antibody titer or their natural immunity. So that all this studying have an inclusion criteria. Probably you guys know that is going to be 1 to 5, 1 to 50, depending on the assay that we are using and depending on the study. But there are two problems with this inclusion criteria. First of all, we don't understand exactly what is the threshold that we cannot that we can prevent dosing of AAV. So the use of 1 to 5 or 1 to 50 has been really chosen based on the lack of lots of information regarding the assay that we are using and lack of a standardization of this assay. So it's important to better understand what is the threshold that below which we can dose safely. And also we conducted already for different conditions some mice study where we use a prolonged use of immunomodulation that prevent the formation of antibody, but also bring that antibody titer below a threshold. So in this objective, we want, of this project, we really want to be able to bring that titer below that threshold and understanding what is that threshold that can be used as safely. So the second objective of this project is really focusing on pre-immunity and allow for all the majority of the kids, even that they are pre-immune, to be enrolled in these trials. So that's the objective of these projects, and I'm happy to answer a specific question about this project.
Dr. Corti, thank you so much indeed. I just wanted, we did have some questions come in beforehand, which we'll go to in a moment, but I just wondered if you could clarify something, because I think when the community started to hear about gene therapy, a lot of us understood that it would be available to all patients regardless of their mutation. And so I think that's one of the reasons why it's attracted so much excitement. But are you basically saying that if these patients have pre-existing antibodies, they will be ineligible for these trials currently? And if you are saying that, what percentage of patients do you and Dr. Byrne think might have these pre-existing antibodies, if that's not too unknown a question?
So far, most of the trial, they exclude subject with pre-immunity. So to answer your question, not all subjects will be eligible with what we use currently. However, the threshold that is being used is not really understood. So we need to do more work understanding what is that threshold. Second, what is the assay that we use to understand that threshold? There are different assays that are being used. As you can see from inclusion criteria of other studies, someone used neutralizing antibody, other people use the total antibody, and they are expressed in dilution. Sometimes they are expressed in a different way. So all this aspect. But so there is, I think, two topics. One, understanding how we measure and what is the threshold. And the second topic is we do believe that we can include more subjects that are included now by using this immunomodulation that bring the titer below that threshold. So far, we saw that more than 50% of subjects will be excluded by this study using this current threshold that for example, the solid trial has been using. So we really want to understand better that threshold, and we do have a management of that titer through immunomodulation, that if we do an immunomodulation for a couple of months before the exposure to AAV, we have some data in the preclinical work in mice that we are able to bring down that titer. So to answer your question, currently, not everyone is eligible for a gene therapy trials, but we are making progress to make this available through this project to a greater portion and hopefully to everyone, but we don't know that.
Okay, thank you so much, Dr. Corti.
If Barry want to add anything on this.
Yeah, Barry, do you want to add something?
You're muted. Sorry. So just to agree, in an overview of this problem, we attempted to find the population of over 200 boys now that have been screened of school age. And certainly there's a relationship between preschool, where these exposures probably happen, as environmental exposures are less in the preschool years, and then certainly towards adolescence, as Manuela pointed out, up to 50% of children reach this threshold for environmental exposure. And we've, one of the assays that we're developing is to determine whether they're in fact specific for AAV9 or any of the AAVs that are being studied, because there are hundreds of different types of AAV, many of which are related to one another. And there's a feature that's called cross-reactivity, where there may be a partial response to one cousin of the AAV that's being used in the study. So we need to understand whether they're specific, whether they represent any safety concerns, as was Manuela stated. And then if so, we want to have a way of mitigating that.
Thanks, Barry. We've had a question, which I wondered if you could expand on. When you mention immunomodulation agents, do you mean plasmapheresis?
No, we actually intend to use agent that a pharmacological agent that induce a B cell depletion. All our experience so far has been based on the use of rituximab and sirolimus. Plasmapheresis does reduce the antibody, but it's very transient. So that's not going to be enough for the viral load that will last at least weeks after the dosing. So the plasmapheresis, we've been doing some preclinical work comparing the two approaches, and plasmapheresis is really transient, so that's not going to work alone. So
Yeah,
it's a pharmacological
a
agent.
very important point to emphasize to the community, because there has been a notion that when the AAV is delivered, it almost instantaneously reaches all the muscle cells. We've been carefully looking at time points early after the infusion, even day by day, understanding that actually the viral vectors persist in the blood for weeks, not just for hours or days. And in fact, the antibodies that would return, as Manuela pointed out, even after plasmapheresis, would be sufficient to eliminate or lessen the effect of the gene therapy vector.
Thank you. So this is a question from Sejal. There are many other initiatives on looking at either immune response or the alternative administration of gene therapy, i.e. using exosomes. And we know that Sarepta's recently announced a partnership with Hansa Biopharma. How does the work that you guys are doing compare to these projects? And will there be any coordination to ensure no duplication?
No, it's also a great question. I don't know that the details of the Sarepta relationship related to exosomes is known. Clearly, there would have to be an efficiency that was equivalent to the AAV administration, and then one would have to determine whether that could be repeatedly administered, because we know that those will not be sustained within cells. And they should be obviously less immunogenic because of the nature of how exosomes are formed from human cells. There are other efforts, as I think you alluded to, that Sarepta is considering other drug therapies, which is a component of the approach we're using. But drugs that only affect one aspect of the immune system and not the cells that make the antibodies themselves are likely to have a lesser impact. And the biggest focus of those treatments has been for liver-directed gene therapy, which we know is not a strategy in Duchenne.
Okay, thank you, Barry. I think we might go to some of our questions that came in earlier before the webinar. And we'll start off with a question about Audentes, because gene therapy has been in the news a lot lately. And last month, Audentes Therapeutics announced that 2 patients had died after receiving AT132, a gene therapy for X-linked myotubular myopathy, using AAV8. How nervous should we be for DMD reading this news?
Well, obviously, this is a tragedy for these families and for the study in particular, and the gene therapy community has taken a lot of appropriate attention to this issue. And there have been some preliminary information that's been shared. We at one time actually were conducting this study at the University of Florida, so we are aware of the approaches that have been taken and some of the non-clinical data. I think one of the lessons that we have to consider here is to pay particular attention to any subtle findings in the non-clinical data, and then make adjustments along the way when human subject data reveals that some of those subtle findings may have represented a concern that needs to be addressed, and you have to be prepared to respond and make amendments to studies based on that. So obviously it is a sad for this study, for the availability of this promising therapy to this patient population, but and it is appropriate to be concerned. And we do think that that's one of the reasons why it's very timely to pursue this current research project, because it's very likely that because of the high dose of vector used in that study, that there is some dysregulation of the immune response that contributed to these findings. And we know that actually in 2 different studies we've done using this approach, that it actually enhances safety because we can manage the immune response and then at an appropriate time restore full immunity through providing immunoglobulin until all of the cells that were suppressed return to full function.
Thank you, Barry. I think the next question is particularly pertinent to the very strange times that we're living in right now. We are in the middle of a global pandemic, at risk of contracting a highly contagious virus, COVID-19. So is this the right time to be looking at drugs and protocols that suppress the immune system?
Yeah, so I provided a more lengthy answer that I can summarize very quickly, is that there's an interesting aspect of those that are most severely affected by COVID-19. They're probably those that have had no prior coronavirus exposure before. And we see some of the same findings, particularly related to complement activation, in those patients who are most critically ill after COVID, after SARS-2 infection. And in fact, that's prompted some investigators, at least in adult patients, to study means of complement blockade. And certainly we know that supplemental IVIG is one of the considered therapeutics to help neutralize this virus. So we're learning really every day about how immunity plays a role in the responses to all viral infections, this one included, and how those 2 things can be balanced to maintain protection against all other real pathogens, and then allow for a treatment opportunity, because certainly time doesn't stand still for those with inherited diseases who really rely on access to gene therapy to try to mitigate that problem. So the studies we're doing now are non-clinical studies. We hope that at some point in the US we'll follow the model in the rest of the EU and see these number of cases decline and even better therapeutic options for those patients. But we don't think that there'll be any interplay between these 2 things in the time frame that this would be considered for patients with DMD.
Thank you, Barry. And then the final question that came in for the webinar. So the week before we announced, Director announced a partnership with Selecta, who developed an immune tolerance platform. Could you just explain briefly what this is, how it could help with antibodies in DMD?
Mm-hmm.
It could potentially be complementary to your program, or is it something totally different?
Yeah, the drug being used in the Sarepta program is a nanoparticle containing rapamycin. And these bionanoparticles actually can aggregate with the vector and provide direct delivery of some of the rapamycin to the cells that particularly take up such macromolecules, like in the liver. And some of the most elegant work with this approach has been done for liver-directed gene therapy, where the ability to get a non-responsiveness to the vector is enhanced when these nanoparticles are delivered. This has been studied at lower doses for gene therapy products that are directed at the liver. We know that may have some benefit in DMD if this observation extends to muscle cells where the genes need to be placed for Duchenne. This same drug is used in the combination we're using, except it's just provided orally. There may be a slightly different mechanism of action, but the end result is the same. I think importantly, what we'll need to determine is when there are higher doses of vector that are given systemically for muscle diseases than for things like hemophilia, for example, where the main target is the liver, whether there'll be a sufficient prevention of the conversion of the antibody producing cells by this drug alone, or whether it will require a combination therapy like we're pursuing.
Thank you, Barry. I think Alex has got a question for you now. Alex Johnson.
Hi, Barry. So one of the questions when I'm talking to parents a lot is the concerns that a lot of the trials for gene therapy seem to recruit relatively young boys in the 4 to 7 sort of age range. Do you expect in the future gene therapy trials to become available for adolescents, for non-ambulant patients? The questions I get asked are why particularly are they using that age group?
Yeah, no, that's an outstanding question, and it partly relates to the regulatory pathway that a given sponsor's chosen. And there's been a lot of emphasis on time function tests that include walking. That's one of the key aspects of why the focus has been on ambulant patients, because if it were possible to demonstrate a prolonged ability to ambulate or improvement in time function tests like getting off the floor, this might be the key outcome measure that sponsors believe the regulators would adopt as demonstrating effectiveness. Obviously, there are important measures of effectiveness for non-ambulant patients as well because of the important influence of cardiopulmonary dysfunction in that age group. So we do think that provided that given the larger total exposure of the vector in older boys and young men, that it will require the same strategy we're studying. So part of what we're attempting to do is make sure there's adequate safety built in to the study plan to allow for older subjects to be included in studies where the focus would be not merely on timed function tests or arm function, but also on cardiopulmonary function, which is obviously critical to long-term health and well-being. So yes, we think this approach will be important for that patient population. Even in the era of commercial approval, it's important to demonstrate a safety strategy for that group of boys.
Thank you, Barry. You mentioned earlier that when you'd screened 200 patients, you found that the preschool exposure was far less. In other words, the younger the boys are, the less likely they are to have been exposed to the virus. What do we know at this point about the efficacy of gene therapy in terms of what age it's given? Do we have any data or any information about that?
Yeah, no, it's an excellent question. This is a double-edged sword. So obviously before there's substantial muscle loss or the inflammatory state worsens within the muscle and it becomes difficult to achieve a correction of muscle cells that actually convert, have that difficulty when they convert to fat. We don't know that is reversible. So in that sense, earlier has advantages. The other side of that coin, though, is that because of the growth expected in children in this early age range of 4, 5, and 6, that will ultimately lead to less durability of the therapy, because as satellite cells divide and repair and grow muscle, they do not carry along with them a copy of the AAV microdystrophin, because they live in a very protected environment at the edge of the muscle cell membrane, and then they divide and increase in number in terms of their ability then to fuse with the muscle fiber. Those two things together actually make it very low percentage of those cells ever see the AAV vector when it's administered. So therefore, they deliver a copy of the set of genes that the individual has that are not corrected. And so the number of instructions that are in the muscle fiber is progressively less and less over time, and that would be expected to lessen the effect. So that's the challenging part of earlier delivery, unless one protects against the antibodies that would allow for re-administration. So to answer your question, earlier administration would be ideal, provided it was done under circumstances where at a later time there could be a re-upping of the total amount of the vector in each muscle cell. and that requires the preventative treatment to block the antibody response.
I mean, Barry, I might put you on the spot, but I know you can handle it because I've seen you at the PPMD conference being asked all sorts of questions. But I suppose one of the torturous things for patients, and something that Alex and I often talk to parents about, is, you know, a real willingness and urgency to try and get onto a clinical trial, even though we know that we are in early stages of this potentially, you know, exciting technology. How realistic do you think it is to hope that gene therapy may be an approved treatment in a few years or even sooner, given your knowledge and experience of gene therapy and Duchenne?
Well, two of the biggest challenges are the topic we're discussing. Make sure that to really be certain that there's safety, we need to manage the immune response in a way that will prevent the adverse events which were described by David in the beginning of the webinar. And then importantly, the manufacturing of the vectors does represent a significant challenge. And I think the good news there is there's been sufficient attention on this problem from pharma and biopharma companies to continually improve the manufacturing process, lower costs, increase access. These are all critical to really making any such product commercially available, because here we are over in the US, we know that there's a reimbursement plan that many companies follow, but that's not shared worldwide. There are many different models for delivery of healthcare, and we have to make sure that these therapies are accessible to everyone, and not just where the reimbursement may be the most favorable. And that's going to require that we continue to innovate on manufacturing. And in fact, it's another aspect of this pre-treatment strategy, that when antibodies are not allowed to rise in the first days after administration, this helps prevent the vector from being cleared out of the bloodstream in a very rapid fashion. In fact, makes better use of the amount that was delivered. So this may ultimately allow a lower dose to be used, and therefore more available to more, if that's a limiting resource.
Thanks, Barry. I think we've got a question now from David Bull, our research director, which relates to CRISPR, which is another treatment approach that we are hearing a lot about in the media. David, do you want to ask your question to Barry and Manuela?
Yeah. Yeah, sorry, I was trying to put this onto the typed in question, but I couldn't make it work. It relates, you were talking about the fact that the muscle cells reproduce, but the copy of the corrected gene doesn't go with them, and so we get this increase. So we get this increase of muscle cells without the corrected gene. So what about the role for CRISPR-Cas9 in terms of genome editing, such that any future multiplication of cells should hopefully include the corrected version?
So at present, and at least to my knowledge, the strategy being pursued by Vertex, the only company that's at least in actively at this very close to clinical implementation of the CRISPR strategy in DMD, is relies on an AAV vector to deliver the editing machinery. Therefore, that machinery will be delivered to the same cells that would be reached by a microdystrophin, for example. Should there be some discoveries that allow that to be preferentially directed towards the stem cells, then there could be an advantage of such an approach, because those cells, as they replicate, would carry with them the corrected copy of the gene. But as it stands now in the current approach, they are not preferentially targeting the stem cells, and the correction is occurring in the myofiber, in the terminally differentiated or mature muscle fiber. That will be subject to the same effects of dilution as the microdystrophins delivered by a similar vector.
Okay.
But certainly the potential is there, as you point out, that if those corrections are made in the very earliest progenitor cells of a muscle fiber, that would be beneficial.
Thanks, Barry, so much. I'm just going to finish off with one last question about the project that we're funding with you, and whether you could just update people on what the timelines are for the project and how quickly we might be able to get some hopeful answers that can help deal with this issue of immunity.
Yeah, well, I mean, it's been really fabulous to work with David and the team at Duchenne UK to finalize the formal agreement that was done very expeditiously. The project's underway in order to establish both these regimens in MDX mice, both here and with a collaborator, and then to actually also see whether additional novel AAV capsids can further enhance the effect, and that this approach is actually universal across a variety of types of AAV, to see how that would play out in the future for a clinical program.
Great, thank you so much. Alex Johnson, I think, wants to say a few words just before we come to an end, but I just wanted to say, Barry and Manuela, thank you both so much. It's been a really informative and useful webinar, and we're just so thrilled to be working with you, so thank you.
Yeah, the pleasure is ours.
No, and just to echo what Emily says, we're delighted to be working with you. We've worked with you on projects before, and we know how diligent and hardworking you both are. And, we're really excited about this project and the potential it has to help bring gene therapy to more patients. And I think, as a Duchenne community, we are constantly watching and following all the latest developments, and, to be partners with yourselves, it's amazing. And, it is a Duchenne UK project. So many charities have helped provide the funding for this. So we just want to acknowledge joining Jack, Duchenne Now and Jacoby's Wish, and all of the family funds who have contributed: Project Go, Cure for George, Smile with Shiv, Team Felix, Following Felix, Team Oscar, Defending William Against Duchenne, Jack's Mission, Chasing's Connor Cure, Help Harriet, Action for Arvin, Backing William and William's Fund. We are incredibly grateful for your support, and we're really excited about the potential of this project. So thank you, everybody, for joining us.
Thank you so much. Bye-bye.
Thank you all.
Thank you.